Have you ever wondered how clean the water is that you're swimming in? Well, I'm out here in Grapevine Lake, Texas to figure just that out. It's a local watering spot that I've taken my family swimming countless times, but how clean is it? So let's do some water quality testing. I'm Dr. Simon, and welcome to my lab. So let's get started first by taking our total dissolved solids from the water. So I've got my nice meter right here. I'm putting it in the water. I'm getting a total dissolved solids of 158. And this meter also is telling me a temperature of 27.8 degrees Celsius, which is feeling pretty good to me. The other reading I'm gonna grab while I'm here is gonna be pH. And I have my pH meter here. And that's a pH of 8.24, which is a little basic, and that's exactly what you want for water. Obviously, you don't want acidic water. Next, I'm gonna grab our first reading that's gonna be important to figure out the biological oxygen demand of the water. And that first reading I'm gonna grab to do that is gonna be the dissolved oxygen that we have in the water. So the waves are lapping a little bit, which is gonna change the reading, but it's stabilized at 6.6 milligrams per liter of dissolved oxygen. So the other thing I wanna do while I'm out here at the lake is grab some water, and I'm gonna also hunt for macroinvertebrates. And those little bugs are gonna tell us exactly how healthy or not our water is. So stay tuned. So I just finished my hunt for macroinvertebrates along the edges of the lake, and man, was I successful. I found everything from larvae to snails to worms to freshwater mussels, some water scorpions, and in the process, I even caught a few fish. I'm gonna catalog each one of these, and then we're gonna take it back to the lab to figure out exactly what they are. Stay tuned. What a morning. We've just come back from the lake, and I've collected some water and some data, as well as a net full of macroinvertebrates. So let's take a look at what we found. So here's what I'll use for this experiment. Obviously, we have our collected water sample that I got from the lake this morning. We also have a water quality test that I purchased online, and that includes everything from coliform bacteria to phosphate and nitrate testing. The additional things that we're also going to need is our dissolved oxygen meter, and that'll allow us to obviously get our biological oxygen demand of the lake. We already took the DO sample from the lake earlier, and so after a 5-day incubation of our water, which I collected also this morning, then we will be able to do a little bit of math and figure out that BOD. So let's talk about these various tests and what to look for when doing each one. The first one is pH, and that's a measure of the concentration of hydrogen ions in water on a logarithmic scale. A pH of 7 is pure water, with acids falling below that and bases rising higher than 7. The EPA suggests that a safe pH for fresh water is between 6.5 and 9. Our water that we measured at the lake was 8.24, so that falls perfectly within that safe range, and it's great for us to swim in as well as safe for aquatic life. Other test that we can do is turbidity. Turbidity is a measure of how cloudy our water is. So looking at the sample we collected earlier to do our BOD testing, we can see that there's some cloudy. But what does cloudy mean? What does a dirty look? Does it have a specific value to do that? To do that cloudy test or turbidity test, you can use a Secchi test. We have one here in our kit. I've already filled this up with water. So when looking at it, you can see our Secchi disc inside, and it looks black and a little bit grayish, which indicates that our water is a little bit cloudy. But what value is associated with that? I think looking closely, it's close to maybe 40 JTUs, which is a mid-range figure for turbidity. Obviously, we'd all rather swim in clear blue water than gross brown water, and I think this one is relatively clear in terms of water goes. Next, we're going to look at nitrates and phosphates, and these two are limiting nutrients essential for plants and aquatic organisms to survive. High levels of these can indicate pollution, and that may be due to fertilizer runoff or even sewage pollution, which can lead to toxic algal bloom. So to do this, I'm going to collect some water. I'm going to grab my beaker, and I'm going to grab some water I collected earlier from the tests. And I'm going to start off with doing the phosphate testing. So we're going to add 10 mLs. Of this, and inside of here is a little tablet, and we're going to shake that until the tablet is dissolved, and then we'll allow it 5 minutes to incubate. So our phosphate tablet is essentially almost all dissolved. Now we're going to give it 5 minutes to allow that to change color. Based on the color change, then we can determine the amount of phosphate that's in our water. While that occurs, we can work on our nitrate tests. It's going to be the same thing, except this time we're going to fill it up with 5 mLs of water. And because this one is light sensitive, we're going to drop it in this silver pouch. We're going to shake it for 2 minutes, and then we'll allow it to incubate for another 5. So our tablet is totally dissolved now, and so we'll allow that to incubate for 5 minutes, and we'll come back to these tests after doing some of the other ones. The next test we're going to be doing is the coliform bacteria test. And this test is going to indicate the presence of toxic sewage and other harmful bacteria that can cause dysentery. For this test, I'm going to do the test now, but we're going to have it incubate for 48 hours. So I'm going to set it up, and then in 2 days, we're going to come back and look at those results. For this test, it's similar. We're going to do 10 mLs of water. And unlike the nitrate and phosphate we just did, we're just going to let this one sit. You don't want to shake it or anything, so we'll allow this one to rest for a little bit. And then, like I said, we're going to leave the lab and come back in 2 days. Earlier at the lake, we collected this sample to measure the dissolved oxygen, or DO, and more importantly, the biological oxygen demand, or BOD. These tests go hand in hand. The DO test determines the amount of oxygen in the water, while the BOD test measures the level of decomposition occurring in the water over time. The concentration of DO is one of the most important factors in determining the overall health of an aquatic ecosystem, as most organisms have a really narrow tolerance range for DO, and most organisms require oxygen to survive. The greatest aquatic biodiversity occurs when DO concentrations are high, so a low DO may signal some not so healthy water. Inversely, a high BOD indicates poor water quality due to the high concentration of organic waste that is being decomposed by bacteria. Typically, high BOD levels lead to low DO levels, which cause dead zones due to hypoxic conditions. The levels we measured earlier at our lake for DO was 6.6, which is a relatively healthy amount of oxygen in the water. After a 5-day incubation, we're going to take this amount of water that I collected, and then we will measure again the dissolved oxygen. Using the numbers that we got from the lake earlier and our results after 5 days, we can measure BOD with a little. And the last thing we measured while being at the lake was total dissolved solids. And that test is used to determine if the water contains a lot of ions or dissolved minerals. The range for this test is relatively large, and depending on the other test we do, it may pinpoint a specific source of pollution. So now it's been about 5 minutes and we're ready to look at the results for our phosphate and our nitrates tests. Let's first start looking at our phosphate test. And so you can see a light blue color. And using the charts that came with our kit, right, we can compare the colors and we can see that our phosphates is about 2 parts per million. The nitrates test didn't really change color, which is good because that shows a nitrate level of about less than 5 parts per million. So mid-level for phosphates and low range for nitrates indicates really not that much pollution in terms of those critical nutrients that plants need. So now that we've completed the tests in the lab, we need to give a little bit of incubation time. Remember, I said our coliform test needs 48 hours and our BOD test needs to incubate for 5 days. So stay tuned because we'll be back for those results. So it's been 48 hours and here is our coliform tests. It started initially clear and now over time the tablet has dissolved and it has turned yellow. The yellow color indicates a positive coliform test, which indicates that there is more than 20 coliform colonies per 100 milliliters of water. This really indicates that yes, there is some sewage or bacteria in the water and that it is definitely not safe for drinking. So this lake water would be one that would be okay to swim in, like we were at our swimming place, but definitely needs additional purification if you want to drink. So now we need to give it an additional 3 days to allow for our BOD test to be concluded. So stay tuned and come back. Okay, so it's been 5 days since we've taken our initial reading from the lake as well as collected our sample to get our final BOD measurements so we can figure out our total water quality of Grapevine Lake. So I'm gonna grab my sample I collected and I'm gonna take my dissolved oxygen meter. And carefully, I'm gonna add that in there. And I'm gonna allow this to kind of level out. And I'm getting a reading of about 5.4 mgs per liter. And so to figure out our BOD, we will take our reading that we took 5 days ago from the lake, so 6.6 milligrams per liter, and then we'll subtract our reading that we just got right now, which is 5.4 mgs per liter. And so this will give us our biological oxygen demand, which is about 1.2 mgs per liter, which is actually a pretty good reading and indicates to us that there is some respiration happening in the lake, but overall there isn't just a massive amount of respiration, which leads to the overconsumption of oxygen. And so it really indicates that our lake is relatively healthy. And talking about the health of a lake, let's figure out the rest of these readings. So overall, I think the biggest indicator of how healthy a lake is will be our DO that we took at the lake, which shows that yes, we have a good amount of dissolved oxygen, which allows life to thrive. We have some respiration happening, but it's relatively low. I think the amount of nitrates and phosphates kind of indicate that there is a little bit of maybe nutrient pollution, but overall not very much considering it's a living and breathing lake. The one thing that was concerning, obviously, was the presence of coliform. And while it's okay to swim in that lake, I definitely would keep your mouth closed when doing it. So the other points that we can look at for the overall health of our ecosystem is our macroinvertebrates. So while we're at the lake 5 days ago, we collected a huge number of different species. And so based on seeing that variety of those species in our lake, we can really show that our lake is living, thriving, and relatively healthy. So let's take a look at some of these a little bit more closely. Macroinvertebrates can give us details about the water quality over long periods of time. I caught a variety of different dragonfly nymphs whose presence alone shows a moderately clean and oxygen-rich water. However, the fact that this one is so large indicates that the conditions of the lake have been moderately clean for a number of consecutive years. Similarly, the presence of freshwater clams and this large damselfly nymph and the additional species of dragonfly nymphs, such as these skimmer dragonflies, also show good water quality for several years. Along the same lines is this caddisfly larva, whose presence is an important bioindicator that the water quality is really good, since they are less tolerant and more sensitive to pollution. While other species are more tolerant to pollution and poor water quality, their presence highlights a large biodiversity, which implies a strong community structure and a healthy ecosystem with minimal ecological stress. For example, the predaceous diving beetle, the water scorpion, and this ferocious water bug can tolerate worse water conditions than the caddisfly, but as top invertebrate predators, their presence highlights a thriving ecosystem with a variety of organisms in the food web. This idea is further reinforced by a variety of other lower food web species that I collected, such as aquatic worms, snails, boatmen, and backswimmers. Overall, these species have a higher tolerance for poor water quality and pollution, but are important players in keeping our ecosystem balanced over time due to their function as decomposers and primary consumers. Because there are always some individuals that can live outside of a species' tolerance range, abundance of certain species helps highlight the predominant condition of our ecosystem. So while I did collect a number of different species, including some fish, I by far collected more dragonfly and damselfly nymphs than aquatic worms or glassworm larvae, which further highlights moderately clean water for at least a couple of years. What an awesome find! This experiment revealed how a little bit of science and a whole lot of time at the lake can give us a clear picture of the health of our lakes and the surrounding ecosystem. So I encourage you to keep the spirit of inquiry alive, look at the world around you with fresh eyes, and never hesitate to ask why or how something works, because every question is a doorway to a new understanding. Lessons like these are what you'll encounter in your APES class, and there's plenty more in our APES course too. Use as a companion to your class to prepare for quizzes and tests and get ahead of your peers on the exam. Thank you for joining us today. If this video sparked your curiosity, please consider liking, sharing, and subscribing. Your support helps us bring more science to life. Until our next experiment, stay curious and keep exploring.